So this is an analysis in the early days based on all the pseudogenes. As I say, the pseudogene resemble a neutral class of loci. So that's why if we analyze the pseudogene, we use them as a neutral marker. But we want to know the mutation bias because there's no functional constraint. So most of the patterns we observe here are contributed by mutation differences. Remember, in the very last few minutes, we talk about there are two things, mutation and fixation probability, right? So these two things contribute to the pattern. Here, the fixation probability is the neutral mutation, like a fixation for the neutral mutations. If any change happen at this locus, doesn't really change the fitness of an organism. So this is the summarized pattern of the changes. And we consider from one state, A, T, C, G, to the other side, right? So if the ancestral state is a G, then we ask, in the pseudogene lineage, what happened? So in this case, it's G to G, so there's no change. But in another case, we observe G to A change. So we can tally, we can count all those changes happen in the pseudogene sequences. And this is how this table summarized. So it look at the ancestral sequence. It's what state is either A, T, G, or C, then change to a different state. So that's the pattern of the substitution. And we can compare the differences showing in this position. And also you can know is the proportion of each size. So the proportion of each size is showing, like from your sequence, is like how many sites are at A states in the ancestral node and how many are in T, C, and G. So this can be counted if we reconstruct the ancestral sequence. We can count those and we can calculate all the changes in the sequences. Then we can get the overall result. So you can show here is you have more ATs and then the GC, so that's a general pattern. So pseudogenes shows more reached in ATs. And that is consistent with our observation. The functional genes, the protein coding genes are slightly enriched at GC sequences. So these are the observation, and we do see the other, like the opposite, the pattern at the pseudogenes. And in the comparison in this type of study has been done in a lot of cases. And this is just one of the case in the early days. They look at 55 pseudogene. Okay. Another thing is I have the word processed pseudogene. Okay, so I'm not quite sure whether we talk about the processed pseudogene. So here the processed pseudogene is the one occur by retrotransposition. So process means the original copy of the gene has exons and introns, but this duplicate copy doesn't have the introns. Why is that reason? Because this gene was originated by the retrotransposition, which means the reverse transcriptase produced by the retrovirus convert the messenger RNA in the cell to a DNA sequence by accident. Actually, the RNA, they want to do their own RNA, put into DNA and insert into the genome. but by chance they pick up a messenger RNA from our cell and convert it into cDNA and insert it back to the genome. They cause a duplication of a functional gene. But it's sometimes or most of the time, this processed pseudogene is not functional. Remember, in order for a gene to be transcribed, we need all the cis-regulatory sequences. So a lot of times when the sequence random insert into the genome, it doesn't have the right promoter to be transcribed. So they sit in the genome and become a pseudogene sequences. So those are the genes analyzed here. It's like a processed, we name it processed pseudogene. So during the RNA processing, so they already chop off all the introns. So here we only look at those type of pseudogenes for this table. But if you hear other people's talk, and because we invite a lot of different speakers, and you know nowadays with increased evidences to know they are processed genes, those retro genes, functional. They are functional. So this set of data is the one we determined or we know they are pseudogenes. And back in that time, all this type of genes be considered as a pseudogene. I'm not quite sure how many of them are true pseudogenes now, or maybe there's one or two has been found not entirely pseudogene, which I don't know because I don't really follow all the genes and the analysis of this gene. But I think in the early 90s, we start to notice those retrogenes can be functional. At least in Drosophila, there are a lot of those retrogenes has been demonstrated. They do have transcription product. They do transcribe in the cell at a certain tissue. So there are some of those retrogenes have tissue-specific expression. So that's just to that you know have to be cautious when you look at those processed pseudogenes. Maybe a small proportion of this are functional at a certain time or tissue. So this is just let you know. We know more about those processed pseudogenes, or the functional one now we call it retrogenes.